Welcome to today's lecture on cell biology and metabolic pathways. In this session, we will delve into the intricate world of metabolism, exploring various processes and pathways that occur within our cells. Metabolism refers to the sum total of biochemical reactions that sustain life and allow cells to function properly. It includes both catabolic reactions that break down complex molecules to release energy, as well as anabolic reactions that build new molecules for cellular growth and repair. Understanding these metabolic pathways is crucial in diagnosing and treating various metabolic disorders. So, let's begin our journey through the fascinating world of cell metabolism. Glycolysis is the first step in cellular respiration, occurring in the cytoplasm of cells. It involves the breakdown of glucose, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon compound. This process generates a small amount of ATP, the cell's energy currency, and NAD, an electron carrier molecule. Glycolysis consists of several enzymatic reactions, including phosphorylation, cleavage, oxidation, and substrate-level phosphorylation. These reactions are tightly regulated to ensure proper glucose metabolism and ATP production. The Krebs cycle is a central metabolic pathway that takes place within the mitochondrial matrix. It involves the complete oxidation of pyruvate, generating ATP, NAD, FOD2, and carbon dioxide as byproducts. The cycle consists of a series of enzymatic reactions, including decarboxylation, oxidation reduction, and substrate-level phosphorylation. The intermediates of the Krebs cycle also serve as precursors for other metabolic pathways. Regulation of the Krebs cycle is influenced by allosteric enzymes, substrate availability, and hormonal signals. Oxidative phosphorylation is the final step in cellular respiration, taking place in the inner mitochondrial membrane. It involves the transfer of electrons from NAD and FOD2 to a series of protein complexes, resulting in the generation of a proton gradient. This gradient is then used by ATP synthase to produce ATP through a process called chemiosmosis. Additionally, oxidative phosphorylation facilitates the regeneration of NAD+ and FAD, allowing glycolysis and the Krebs cycle to continue. Various factors, such as oxygen availability and the electron transport chain's integrity, influence oxidative phosphorylation. Lipid metabolism encompasses the breakdown, synthesis, and modification of lipids in cells. Fatty acids derived from dietary intake or adipose tissue triglycerides serve as the primary energy source during fasting or prolonged exercise. The beta oxidation pathway converts fatty acids into acetyl-CoA molecules, which can enter the Krebs cycle for ATP generation. On the other hand, lipogenesis involves the synthesis of fatty acids from excess dietary carbohydrates. Lipids also play essential roles in cell structure, signaling, and insulation. Protein metabolism involves the breakdown and synthesis of proteins in cells. Proteins are essential for various cellular processes, including enzymatic reactions, transport, signaling, and structural support. Protein catabolism occurs through proteolysis, where proteases break down proteins into amino acids. These amino acids can then be used for ATP production through gluconeogenesis or participate in other anabolic pathways. Protein synthesis, on the other hand, is regulated by transcription, translation, and post-translational modifications. Amino acid metabolism encompasses the degradation and synthesis of amino acids in cells. Amino acids serve as building blocks for proteins but also play critical roles in cellular signaling, neurotransmission, and energy production. Catabolic reactions, such as transamination and deamination, break down amino acids into intermediates that enter the Krebs cycle or gluconeogenesis. Anabolic reactions involve the synthesis of non-essential amino acids and the conversion of one amino acid into another. Nucleotide metabolism involves the synthesis and recycling of DNA and RNA nucleotides in cells. Nucleotides serve as essential components for DNA replication, transcription, translation, and cellular signaling pathways. De novo synthesis of nucleotides occurs through several enzymatic reactions, starting with sugar and base synthesis and ending with nucleotide assembly. Salvage pathways also recycle nucleotide bases from degraded DNA and RNA molecules. Imbalances in nucleotide metabolism can contribute to genetic diseases and cancer. Metabolic pathways are tightly regulated to maintain cellular homeostasis and respond to changing physiological conditions. Regulation occurs at multiple levels, including enzyme activity, substrate availability, and hormonal control. Allosteric regulation, covalent modifications, and feedback inhibition are mechanisms employed to ensure proper metabolic flux and prevent wasteful energy expenditure. Dysregulation of metabolic pathways can lead to metabolic disorders like diabetes, obesity, and metabolic syndrome. Cells have the ability to adapt their metabolic pathways to respond to changes in nutrient availability, energy demand, or stress conditions. Various signaling pathways, including insulin signaling and the AMP-activated protein kinase pathway, help coordinate metabolic adaptations. These adaptations can include switching between different energy substrates, altering gene expression, and modulating enzyme activity to maintain cellular viability. While glucose is the main carbohydrate fuel for most cells, other carbohydrates can also be metabolized for energy production. Galactose and fructose, for example, undergo specific enzymatic reactions to enter glycolysis or the Krebs cycle. Additionally, polysaccharides like glycogen and starch serve as storage forms of glucose and are broken down when energy demands increase. Understanding the metabolism of alternative carbohydrates is essential in certain disease contexts, such as galactosemia or fructose intolerance. Metabolic disorders arise from abnormalities in cellular metabolism, often due to genetic mutations or environmental factors. Examples include diabetes mellitus, phenylketonuria, and lysosomal storage diseases. These disorders can affect various metabolic pathways and result in a wide range of symptoms and complications. Diagnosis and treatment of metabolic disorders often involve lifestyle modifications, dietary interventions, and targeted pharmacological approaches. Different cells and tissues in the body have unique metabolic requirements and regulations. For instance, the liver plays a central role in maintaining glucose homeostasis through the processes of glycogenolysis, gluconeogenesis, and glycogenesis. Adipose tissue is involved in lipid storage and release, while muscle tissue utilizes energy sources during physical activity. Understanding how metabolic pathways are tailored to specific cell types and tissues is crucial in managing metabolic diseases and optimizing therapeutic interventions. In conclusion, cell metabolism encompasses a series of interconnected pathways crucial for maintaining cellular function. Familiarity with the regulation and adaptations within these pathways is vital for diagnosing and treating metabolic disorders. By understanding these complexities, medical professionals can identify potential therapeutic targets.